Early evolution of the eye. The earliest predecessors of the eye were photoreceptor proteins, specialized cells containing two types of molecules in a membrane. The opsin, a light-sensitive protein surrounding the chromophore, a pigment that distinguishes colors. Groups of such cells are termed eye spots. Eye spots are found in nearly all major animal groups and are common among unicellular organisms, including Euglena. The Euglena's eye spot, located at its anterior end, is a small splotch of red pigment which shades a collection of light-sensitive crystals. While the early eye spots permit animals to gain only a very basic sense of the direction and intensity of light, the development of an optical system that can discriminate the direction of light to within a few degrees is apparently much more difficult. Such complex optical systems started out as the multicellular eye patch, gradually depressed into a cup, which first granted the ability to discriminate directional brightness, then in finer and finer directions as the pit deepened. While flat eye patches were ineffective in determining the direction of light, as a beam of light would activate exactly the same patch of photosensitive cells, regardless of its direction. The cup shape of the pit eyes allowed limited directional differentiation by changing which cells the lights would hit, depending upon the light's angle. Pit eyes, which had risen by the Cambrian period, were seen in ancient snails, and are found in some snails and other invertebrates living today, such as Planaria, though still much more useful for detecting the absence or presence of light than its direction. This proto-eye gradually changes as the eye's pit deepens and the number of photoreceptive cells grows, allowing for increasingly precise visual information. During the Cambrian explosion, the development of the eye accelerated rapidly, with radical improvements in image processing and detection of light direction. The primitive Nautilus eye, functioning similarly to a pinhole camera, was developed as the pit deepened into a cup, then a chamber. By reducing the size of the opening, the organism achieved true imaging, allowing for fine directional sensing and even some shape sensing. Lacking a cornea or lens, eyes of this nature provide poor resolution and dim imaging. But, for the purpose of vision, they are still a major improvement over the early eye patches. Lenses evolved independently in a number of lineages. Simple pit eyes probably developed lenses to improve the amount of light that reached the retina. The focal length of an early lobopod with lens containing simple eyes focused the image behind the retina, so while no part of the image could be brought into focus, the intensity of light allowed the organism to see in deeper and therefore darker waters. A subsequent increase of the lens refractive index probably resulted in an in-focus image being formed. The development of the lens in camera-type eyes probably followed a different trajectory. The transparent cells over a pinhole's eye's aperture split into two layers, with liquid in between. The liquid originally served as the circulatory fluid for oxygen, nutrients, waste, and immune functions. Yet, it later turned out that the multiple interfaces between solids and liquids would increase the optical power, allowing wider viewing angles and greater image resolution. Eyes later have gone through a wide range of adaptations to meet the requirements of the organisms which bear them. They vary in the range and wavelengths they can detect, their sensitivity in low light levels, their ability to detect motion or whether they can discriminate colors. As opsin molecules were subtly fine-tuned to detect different wavelengths of light, at some point, color vision developed when photoreceptor cells developed multiple pigments. Similarly, night and day vision emerged when receptors differentiated into rods and cones respectively.